Hey guys, Mr. Nichols here, looking to review the vast majority of our concepts that will be appearing on the final exam here for the fall semester for biology. So, for those of you in Mrs. McElroy's class, hello. I'm going to be here kind of hopefully summarizing a lot of the things that you have talked about. Me and Mrs. McElroy were pretty well aligned with the way we kind of explain things and how we kind of covered things in our previous ecology unit exam and some of our assessments. So I'm assuming that most of this is going to sound pretty familiar. If anything kind of stands out to you, feel free to reach out to me. I am a teacher on your Google Classroom account, so you can always reach out to me there. You can also reach out to Mr. Lasella, your guys' sub here. So I did want to kind of go through and focus in on some of the parts that are going to be a little bit more important for the short answer section first. So the short answer section of the final exam is going to be worth about 70% of your overall grade on the final exam. So the multiple choice section is only going to be worth 30%. So this short answer section on your study guide is really important to make sure that you're pretty confident with it. Now, before we go ahead and dive into this, I think it would be good for us to take a moment and just kind of review the different parts of our CER structure, right? So we have claim, evidence, and reasoning. Now, you won't be constructing really any claims, but you will need to create an evidence statement and also provide some reasoning. So we need to definitely identify the differences between each of these first. So anytime we have a scientific question that we're going to try and answer, your claim is what you think the answer to that question is. So oftentimes for this final exam, that's going to be given to you. Next semester, we're going to start to work on constructing the full process all the way down to the bottom. But again, for this final exam, we're just working on evidence and reasoning. So the big key important part here is going to be the evidence and the reasoning. So evidence must be just numerical data that's shown in a graph or a table or a chart, some sort of study that's been done. Now, we need to have our evidence be the backbone of what we're trying to say. If we just make a claim with no evidence, it's not going to be a claim that's very valid and very strong. It's not going to be a strong argument that you can make. So we need to have evidence to back up anything that we say. So in our cases, the evidence is going to just be the data that you see that's relevant. We don't want to just list all of the information that's in there, but we want to pick out what evidence and what numerical data would be important for our argument that we're trying to make. So the one big thing and the number one issue that I see people struggle with is I think we have this innate desire to just explain why things are, just because, we want to try and get the right answer, right? So a lot of times if we're not 100% certain of ourselves, we just throw a bunch of extra info in there. I am going to ask you guys to not do that for your evidence statements. Your evidence statement should not have any sort of an explanation in it. It's literally just, here is my data. That's it. So the explanation comes in the reasoning section. So anytime we ask you to say, explain your thinking, or this happened because, that's going to be the reasoning section. So the reasoning is going to explain how or why your evidence supports your claim. So why is it that your evidence backs up what you're trying to say? So that's what we're going to be looking at here. So we have a couple of examples on our study guide of this. So for example, the short answer section here. So we've been given, for this first one, we can see an energy pyramid. So we can see the amount of energy that's available at each of these different trophic levels. So notice it decreases in a pattern, right? So that's kind of like our data table here. So the claim in this case that we're trying to support is that 10% of the energy transfers from a lower trophic level to the next trophic level up. Basically, when an organism eats something, it's only able to gain access to 10% of the energy that's stored inside of whatever it ate. So a good evidence statement here, remember, evidence is just going to be the numerical values. So what you could do here is we could say that the primary producer here, or sorry, primary consumer, that's going to be our grasshopper, is going to eat the grass. And the grasshopper is only going to gain 60,000 calories of 600,000 calories. And then you could extend that even further and say that this is, so 60,000 calories is 10% of 600,000. And that's it. You're only just saying the numbers, the values that you're dealing with here. So that would be a good evidence statement of what we'd be looking for here. Okay, so now number 2 gets a little bit trickier because we have a data table that's showing a change over time, right? So if we look here, the claim in this second one, which this is our nitrogen cycle aquarium example. So the claim is saying that a large decrease in the Nitrosomus bacteria, which is this one right here. So the Nitrosomus bacteria population affected how nitrogen is cycled in the fish tank. So what we have to do is we have to show how that change occurred, or provide some data that shows how that change occurred. Now, if you look here, the Nitrosomus bacteria, it's connected to both ammonium and nitrites here. Now, technically, the graph down here, or the chart down here, says ammonia. That was a little bit of a typo. That should have said ammonium, but that's okay. So when we're looking at this here, essentially what we need to do is we need to choose some pieces of evidence that explain how the Nitrosomus bacteria impacted our aquarium. So we have to show that change over time. So what I would like you to do, and what I would imagine Mrs. McElroy would have wanted you guys to do, is to choose 2 dates on here and basically say how the ammonia or the nitrite changed. We don't really want to say the Nitrosomus bacteria population because that's already what we said in our claim, right? So we want to go ahead and use some of these other pieces of information. So a good evidence statement here would be that saying that between 927 and 929, the ammonia concentration, the ammonia amount, whatever you want to say there, increased from 15 to 50 ppm, or what stands for parts per million. You don't need to know that part too much, but all you really need to know is that you just said what the change was. So the ammonia concentration increased from 15 to 50 parts per million. You could do the same thing for nitrite. So you could say between the dates of 927 and 929, the nitrate levels decreased from 60 ppm to 10 ppm. That would be totally fine. Now notice for both of these questions, there's no explanation of why that is, because we're not asking for a reasoning statement here. You will most likely get deducted points if you go through and explain in this answer, for example, why this evidence supports your claim. We're not looking for that here. All we care about are just the numbers. We should not be having any sort of reasoning or explanation going into this part here. Okay? All right, so that takes us further into kind of question 3 here. So looking at this, okay, we're looking at a change in the biodiversity. So the biodiversity of this forest ecosystem started at 0.2, and then it ended at 0.012. Okay, so the question's asking, what do you think might be the cause of this change? So what we would need to realize is that the biodiversity is decreasing. So biodiversity is going down. So that means that something negative must be happening to that population, to the organisms that are living in this forest ecosystem. So there's a wide variety of things that you could put here, but things like loss of habitat, loss of food, those would be possibilities here. You could also say things like increased hunting or more foot traffic from people. So there's a lot of different things that you could put there. And as long as you provide some sort of an explanation, a logical explanation of why or how you think that those factors impacted the change in biodiversity, that's going to be fine with me. Okay? All right, so this next question here, looking at the black-tip sharks, the cow nose rays, and the sea scallops. So basically, and I'm not going to go and read through all this here, but basically what this is getting at is that the sharks eat the cow-nose rays and the cow-nose rays eat the sea scallops. So they all have a relationship where they interact with each other. Okay, so this section that shows says the data showed that, this is essentially our evidence statement. So you should not have any explanation here. All you're doing is you're just saying what's happening to each of the populations here. So the blackfin sharks are decreasing, the cow-nose rays are increasing, and then the sea scallops increased, and now they are decreasing. Okay, so now this part here where it says this happened because, this is essentially our reasoning statement. So the reasoning statement here is explaining why you think that those changes are happening. So something along the lines of the blackfin shark their population was decreasing due to them being hunted for shark fin soup, and because there were less sharks eating the cow-nose rays, the cow-nose rays population increased, and then because there were more cow-nose rays, then the sea scallop population was starting to decrease there. Okay, so something along those lines is exactly what we'd be looking for there. Okay, so I would argue that this last one is a little bit more tricky, and it most likely will be the trickier one of this short answer section on the short answer part of the final exam. So I would definitely make sure you have a pretty solid understanding of this. So the relationship that we're trying to explore here is basically thinking about cellular respiration. So in the yeast respiration lab, we had a sample of yeast, we gave them some sugar, and they went through cellular respiration. So we have to think about what comes out of cellular respiration. Okay? So we'll get back to kind of our rationale here in just a moment. So the scientific question's asking, which type of sugar will produce the highest amount of ATP in the yeast? Now, if you think about it, we're not measuring the amount of ATP in the yeast. But if you think about what's being put into this balloon, and we think about the products of cellular respiration, right? If we think about the equation for cellular respiration, the products are going to be carbon dioxide, water vapor, and ATP. Now, each of those are going to be independent of each other. So as we have more cellular respiration taking place, we're going to get more carbon dioxide, but we're also going to get more ATP being produced. So a good claim to follow this, if we look at our data table here, okay, is that glucose will produce the highest amount of ATP, because if we look here, it produced the most CO2. So that's kind of our rationale for why that is the case. Now we got to kind of expand on that a little bit further. So, all right, so looking at this next section here, where it says to provide 3 pieces of evidence, that's going to be, basically you're just picking out 3 data points from our data table here. So you definitely want to pick glucose, because that's definitely the highest amount of CO2 that was being produced. And then you can choose any other 2. You can choose the control down here with 0 centimeters. You can choose the lactose with 1 centimeter, or you can choose the fructose with 35 centimeters. Okay. All right. So now kind of expanding on that, our data showed that, again, this is now our evidence statement, even though I probably should have reformatted that because technically the reasoning is this part right here. So our data showed that the glucose was the largest balloon because it produced, or sorry, I should back that up. So the glucose had the largest balloon, fructose had the second largest, lactose had the third largest, and then the yeast and water didn't have any CO2 that was produced. So they had the smallest balloon circumference. So now for our reasoning, this happened because the yeast converted that glucose and produced the most carbon dioxide. So since ATP is another product, another output of cellular respiration, then more ATP would have been produced with the glucose as well. If we produce more carbon dioxide, then we're going to produce more ATP. So that's kind of our relationship there. Okay, so again, this is going to be probably the most difficult question on the short answer section. The rest of it is pretty straightforward, and it's pretty similar to what we saw in our ecology unit exam. Okay. All right. So I will quickly go through a lot of this multiple choice section. I do want to let you guys know, though, that both Mrs. Mack and myself worked really hard to come up with some multiple choice questions that were pretty difficult. We intentionally made them more challenging because since we're taking these tests in remote environments, we need to make sure that you guys can't easily Google them. I know there's a lot of temptation to do that. So we specifically designed these questions so you wouldn't be able to just Google the answer and then just copy and paste the response in there. That's just not a very meaningful way of taking a test. So what our multiple choice questions are going to be getting at some of the core concepts. You will have to kind of think a little bit. So I strongly recommend that you take your time. It's really not going to be very long, and you're going to have the whole class period, whatever day you are taking that part of the final exam, to go ahead and go through it. So please take your time and really think carefully through your responses. So the study guide is kind of highlighting the general answers to these. So the questions on the final exam will be a little bit more in depth than these ones are. So I'm not trying to say that to be mean or anything like that. I just want you guys to be prepared so you're not sitting there when you take the final exam being really blindsided by these types of questions. So just keep that in mind. You need to have a deep understanding of these concepts. Okay. All right, so let's just kind of talk through this here. So the equation for cellular respiration, pretty straightforward, right? We've got glucose, so C6H12O6 plus oxygen is going to react to yield water, carbon dioxide, and then ATP. Okay, so at this point, we should be feeling pretty confident with that. Another two terms you should have a pretty good idea of. So we've got the reactants on the left side and the products on the right side. So products, think of what's being produced. So products are what's coming out of the reaction. Reactants are what are going into the reaction. Okay, so the purpose of cellular respiration is to convert glucose into usable energy. So remember, glucose is kind of the stored form of energy. The usable form of energy is ATP. So cellular respiration is going to release the energy that's stored inside of the bonds of glucose in the form of ATP. Okay. So this right here, I would say that this is a pretty big picture idea. This was the biggest thing that Mrs. McElroy and myself wanted you to walk out of our small cellular energy unit with. So the idea here is that if we look at both the cellular respiration equation and the photosynthesis equation, that they both lead into each other. So the products of photosynthesis are the reactants of cellular respiration. The products of cellular respiration are going to be the reactants of photosynthesis. So it's this connection between each of these different processes that makes them require each other. So photosynthesis needs carbon dioxide and water in order to take place, and cellular respiration needs oxygen and glucose to take place. So what we end up with is this cycle, this repeating cycle that the outputs of one process feed into the inputs of the other process. So that's kind of our big picture idea here. Okay. All right. So going back to our study guide here. So cellular respiration obviously takes place inside of the mitochondria. That's a pretty quick one there. Excuse me. So what forms of energy do you start with and end with in cellular respiration? So in cellular respiration, you're starting with glucose. That's the stored form of chemical energy. And you're ending with ATP. That's the usable form of energy, okay, that your cells can actually use. Okay. All right, so for the yeast respiration lab, be able to analyze a graph. So if you need to look back at your yeast respiration lab, look back at the graphs from that. So the just kind of broad strokes here, the independent variable for that lab was the concentration or the amount of sugar that was in each of the samples. So the dependent variable is what we're measuring, right? So it was the amount of carbon dioxide that was being produced. So we can actually go back up into this one, which this one's a little bit different because in this one, the independent variable was the type of sugar. And then the dependent variable was the circumference of the balloon, or the amount of carbon dioxide that was being produced. So remember, the independent variable is what I am changing as the experimenter, and the dependent variable is what we're trying to figure out, what we're trying to measure, what's going to change there. Okay. All right, so looking back to the plant and snail lab, which, to be honest, I don't think that Mrs. McElroy's class went through and completed that. So you guys don't need to worry about these questions on the final exam. They won't show up on the final exam. I'll make sure that they aren't on there for you guys. But so for the plant and snail lab, okay, there was BTB, which was bromothymol blue, and that's an indicator that changes color based on the presence of carbon dioxide. Basically, when the acidity changes, but we don't need to know that too much. Basically, what we need to know is when there's a lot of carbon dioxide in BTB, the color changes from green into yellow. When there are low amounts of carbon dioxide, it changes from green to blue. So what we saw in this lab was when we just had plants present in our sample, the BTB changed from green to blue because those plants were taking the carbon dioxide out of the liquid and then adding more oxygen. So the yellow color was when we had the snails. So when we had the snails, they're not going to be producing oxygen. They're going to be just producing more carbon dioxide, and they're going to be taking the oxygen out of that test tube. So that would cause the color to change to yellow because they're going through cellular respiration. So that's kind of our big picture idea there, which if you think about it, if you mix paint together, if you combine yellow and blue, you get green, right? So that's kind of where the green comes from when they're both combined in there. Okay, so we already kind of looked at this. So photosynthesis. So we've got 6 water molecules and carbon dioxide combining together with light as well, which I'm actually going to add that there. Okay, to produce glucose and oxygen. So again, those are required for cellular respiration to take place, right? So the purpose of photosynthesis is not to make oxygen. That's not the goal of plants. It's just a byproduct, even though we really need it in order to survive. But the purpose of photosynthesis is to make glucose. Okay. All right, so moving on. We already kind of talked about some nature of science stuff with the yeast lab question when we were talking about independent and dependent variables. So moving into ecology here. So if we're trying to calculate the biodiversity index, remember we're just going to take the total number of different species and divide it by the total number of organisms. So in this case here, we have 8 different species, 40 total organisms. So we're just going to divide 8 by 40, and that's going to give us 0.2. So basically anything below 0.4, we're going to call that low biodiversity. Anything between 0.4 and 0.7, we're going to call that relative biodiversity. And then anything 0.7 and above is going to be high biodiversity. Okay? And here's the general idea that the closer we get to 1, the higher the biodiversity is going to be. Okay? So this one's looking at trophic levels. So here we've got the trophic levels of the deer. So this first one's saying that we have 1 down here with looks like some shrub or tree, 2 and 3 with the mountain lion here. Okay, moving over to the robin. The robin would be, if we're following that chain, is going to, if we look here, the grass is the producer. The caterpillar is going to be the primary consumer. So the robin will be the secondary consumer here. Okay, and if we look at the fox, And I apologize if this question wasn't very clear. What we were assuming is if the plants down here, the grass, gets 100% of the energy, how much of that 100% would be available to the fox. So according to the 10% rule, it goes down by 10% each time. Sorry, 10% is only left over at each time. So if this is 100% for the grass, the caterpillar would be 10%, the robin would be 1%, and the fox would be 0.1%. Okay, so looking at this next one here, it's asking what's going to happen to each of these different organisms if a disease came along and almost completely wipes out the mouse population. So if we think about this here, the mouse is eating the grass, so the grass population will increase. And if we think about this here, okay, so if the grass population is going to increase, then the rabbit population is going to increase. So then the mountain lion population is going to increase as well because they're going to get more food. So the caterpillar is also going to increase here. Okay. And then, so if we look here, because there was more grass, caterpillar increases. So for the hawk, because the hawk feeds on the mouse, that means that they've lost a fair chunk of their food that they're going to be eating. Now, they'll still survive because they can eat the rabbits here too, but their population is most likely going to decrease there. So that's a good example of how we need to know population change. Okay, so looking at this one here, so here we've got some seals, and we have got a population of seals over the past, or between 1975 and the year 2000. So if we look there, when we're trying to figure out the carrying capacity of the seal population, instead of looking at just the highest point here, we need to look at the highest part of this curve. So that's the maximum number of organisms that can live in a particular area. So if we look here, which looks like this answer key needs to change because it's closer to 7,500 here. So if we look, the very peak of this curve is going to be our carrying capacity. That's the maximum number of organisms that can be sustained in this area. Okay. So looking at this here. Okay. So moving on to succession. So if we think back to what we talked about with primary versus secondary succession, primary succession means that we're starting from scratch. So if a volcano is erupting in the Pacific Ocean and it creates new land, there's not going to be any soil on it, right? There's not going to be any living organisms on there. It's just rock. So primary succession takes place when there is nothing there present on the ground already. So the types of organisms that are going to colonize this area are going to be pioneer species. So these are going to be things like lichen and mosses. Things that are going to be carried by the wind to this island. So now let's say we're talking about a forest fire. So a forest fire, like the ones we've seen this year, comes through and wipes out an entire forest. There's still going to be soil on the ground underneath where that forest fire took place, right? So basically, because this is secondary succession, the living organisms that recolonize that area aren't going to need to start out from scratch. They already have soil that's present on the ground. So it's going to take that land and that area, that environment, much less time to get to that climax community where we have a very healthy environment with a rich array of different organisms. Whereas the volcano example, that's going to take much longer because we have to develop all of the soil that's present for, flowering plants and trees to be able to grow on that island. So that's a key difference between primary and secondary succession. So last but not least here, so we have, these two graphs where we're trying to identify the trend that we can see between both of them. So this first graph is a temperature anomaly graph. So basically showing how it's changed over time. So basically what we're seeing here is that the change in average global temperature every year has been increasing since about 1950. And there are some slight fluctuations. It does kind of dip, but the overall trend is that the temperature change, that temperature anomaly, is increasing every year. So for the second graph here, notice how the amount of carbon dioxide levels that was stored in these ice cores, that's where these are from, that's why we can go back hundreds of thousands of years, way before humans were able to take any sort of scientific measurement. We can see that there's a steady kind of pattern here where we have, a significant decrease in carbon dioxide, then a significant increase, and then a significant decrease, and then a significant increase. So our planet has kind of followed this pattern historically for millions of years until recently. So if we look here, about the last 50 years or so, we've seen a dramatic increase in the amount of carbon dioxide that's being produced here. So the big thing that I want you to try and tie here together is we don't necessarily know that these are 100% connected. There's lots of data sets that kind of line up that might not necessarily be connected. But if we look here, there's a pretty consistent trend. If we look starting at 1950, which would be about here, until now, there's a pretty consistent increase in the global temperature change each year and the amount of carbon dioxide in the atmosphere. So that's kind of the relationship we should have put together with those here. Okay, so I'm hoping that this served as a constructive review for you. I'm hoping that if you have any questions, you please don't hesitate to ask myself or Mr. Lasella for Mrs. McElroy's class. So keep in mind that we're going to be splitting this test into 2 parts. So remember, the short answer is going to be next week. So for our Blue Day classes, that's going to be Monday and Tuesday. And then for our Silver Day classes, that's going to be Wednesday and Thursday. So the multiple choice section will be on Friday for Blue Day classes, and then Monday, the 21st, for Silver Day classes. Okay, so make sure that you show up to your Google Meet for each of those class periods. That's the only way you're going to be able to take the final exam. So make sure to ask any questions as they come up. Wish you all the best and good luck. You guys got this, and take care. Thanks for stopping by.